Okay, let's get started. And today we'll talk about DNA repair and recombination. And this is a very interesting and quite complicated topic. And I'll give you a brief overview about various kind of mechanisms for DNA repair. And by the end, we'll talk a little bit about the DNA recombination. And this is a very important process. And for example, it is involved in the generation of the different kind of antibodies in our body. And we know we have a very large genome. In human cells, we have 3 billion base pairs of DNA. And every time when the cells divide, that 3 billion base pair of DNA have to be copied accurately. And in the process of throughout our life, the DNA have DNA have to be maintained so that there's no, if there's damage, it can be repaired. And you see here are some of the potential causes of DNA damage. For example, chemicals. We are constantly exposed to various thousands kinds of man-made chemicals. A lot of them can cause DNA damage. And also, we are constantly exposed to various kind of radiation. For example, ionizing radiation from the universe, and also the light from the sun, the ultraviolet radiation from the sun. It can also cause DNA damage, and cellular metabolism, which sometimes also cause DNA damage, and of course. The replication process may have errors, and it's unavoidable because the genome is so large. So this various kind of damage to the DNA can actually cause various kind of response. And for example, it can cause the cell cycle and checkpoint activation, and also, for example, transcription activation. And sometimes if the DNA damage is really bad and the cells cannot repair it, and then those cells will get rid of. So they will go through this apoptosis process, the programmed cell death. Those cells will be eliminated. And in a lot of cases, damage to the DNA can be repaired. That's what we'll talk about today. And those involves direct reversal and a base excision repair, a nucleotide resection repair, a mismatch repair, or even double-strand break can be repaired, for example, by homologous recombination or by direct error-prone double-strand end repair. So first here, I introduce you to one of the kind of DNA damage which is caused by ultraviolet radiation of the DNA, and two successive Simon base in a DNA can be cross-linked when they are exposed to ultraviolet light, and they will form this. You see, 2 bonds between those 2 6-membered rings. And this kind of bond formation will cause a big deformation in the structure of DNA. And of course, it will affect, for example, transcription or replication process. And so, in the cells, they have this DNA photolyase. This is the enzyme which can actually recognize those cyclobutyl ring of the Simon dimer. And then it will break those 2 covalent bond and restore the Simon bases. So also we know there's a lot of chemicals, reactive chemicals that can react with the DNA and sometimes even modify the DNA. And for example, this cytosine can react with nitrous acid to form uracil. So this uracil will form a base pair with adenine instead of cytosine. Like originally, it should form base pair with thymine, but if there's a modification, it can form a base pair with adenine. And also, the the amine group on adenine can be removed, modified by nitrous acid to change into this into a hyperxanthine, and which conform base pair with cytosine. So these are the chemical modification of the bases, and this like if you look at the foods, like especially preserved meat, in a lot of cases, and you'll see some of them contain nitrates. And under acidic condition, nitrates will generate this nitrous acid. It can modify the DNA. So this is a food preservative. I believe it's still used in a lot of products. And if it is in a very low level, it should be okay. But if it is high level, if you consume those food like in a long time, they may cause DNA damage, which eventually may lead to cancer. So this is the nitrate cause nitrous acid, nitrate, they cause cancer if you have too much of those. And also there are various other reagents. For example, this dimethylnitrosamine, and this is a pretty reactive molecule. It can modify the base, the guanine base, and add a methyl group to the 6 position oxygen. And this modification of a guanine, you see it, the unmodified guanine confer base pair with cytosine. But when this is modified by this 6O6 methylation, and then it conform base pair instead with thymine group, thymine base. And you see now the original GC base pair now changes to a GT base pair. And eventually, after several rounds of application, that original GC base pair eventually changed into an AT base pair. And so with this one base change, it may change the identity of the amino acid, which is coded by that segment of the gene. So methylation of the guanine bases also result in mutation. And so we have in our cells there. There's a repair mechanism, which is a family of enzyme called methyltransferase. And this enzyme actually specifically repaired the O6 methylguanine DNA of the base. And so this enzyme, it have in the active site, it have a cysteine residue, and it can react with this modified methylguanine to remove that methyl group. And so you see, at the same time, once this enzyme got modified, its active site is reacted with a methyl group. And it can only do one round of reaction. It is a very expensive process to fix this modification of the guanyl nucleotide. So sometimes DNA damage can be repaired by the base excision process. For example, if this base is damaged, and then there's enzymes called DNA glycolase, it will remove the base from the ribose. And then the endonuclease, which specifically recognizes a basic region, and it will cut open the first DS bond, and then the enzyme will actually remove a small stretch of the DNA. And then the DNA polymerase will come in, it will synthesize a new piece of DNA, and eventually the DNA ligase will connect those fragments together. And you see from here to here, that damaged base is replaced. And it's not replaced by removing that one and putting a new one, but it is replaced by removing that nucleotide and a segment after it, a short segment of nucleotide after it, and then synthesize a new segment and then connect them together. So that is a DNA repair by base excision. And so this is another mechanism of base excision, nucleotide excision. In some cases, when there's a damage, which is pretty pronounced, and it causes major deformation of the DNA. And then, like you see here, in both human bacteria, they have enzymes which can recognize those damaged DNA, and then cut the DNA phosphodiesterase backbone and 2 points upstream and downstream of that segment of DNA. And then next DNA helix come in, and it will remove that piece of DNA. And then DNA polymerase 1 come in, and it will put in a new piece of DNA. And then eventually the DNA ligase connects the fragments together. So you see that damaged part can be repaired. And so the mammalian cell system have slightly different mechanism, and because those enzymes cut at different places, upstream and downstream of the damaged point. And it will remove 20 nucleotide segment of the DNA, and then DNA polymerase S epsilon. It will come in and synthesize new strand, new segment of the DNA. And eventually DNA ligase will come in and connect the fragment of DNA together. So this is it's pretty similar to what we show you here, and but it is a slightly different kind of mechanism. You see it cut upstream and downstream, and then remove that segment. And then the DNA polymerase will come in and put in another new strand of DNA. So in bacteria, the DNA replication process actually involves a modification of the DNA. And you see, these are the original template DNA. They have a methylation at a certain adenine base in this GATC sequence. And when the DNA got replicated, and the newly synthesized strand, even it contains this GATC, and it is not modified. So at this stage, the cell cannot distinguish between which one is the original template and which one is newly synthesized. And after a certain period of time, and then those sequences can be newly synthesized DNA can be recognized and modified. And then the cells cannot distinguish between the two strands. So in our case, if there's a mismatch, which happened in the replication process, and then at this stage, the mismatch can be recognized, and then the DNA, that mismatch can be repaired. Because at this stage, the cells can recognize which one is the original, which one is the newly synthesized. So this family of enzymes called MUT-S, they are involved in the recognition of the mismatch. And you see, these are the structure of the MUT-S DNA complex. And it recognizes the DNA, and you see MUT-S and MUT-L bind DNA at the mismatch region. And then you see that the DNA actually loop around this enzyme, and then the mismatch region now come out of form a loop out of this this enzyme. And then the MUT-H enzyme is recruited, and it will cut, this is a nuclease, it will cut the DNA, the newly synthesized strand of DNA, and this unmodified GATC sequence. So once that cleavage happens, and then the cell have a new helices and the nucleus, they can come in and they actually remove a stretch of DNA. Eventually, DNA polymerase 3 come in and it synthesize the strand, and then you see now the mismatch is repaired. And then the fragment can be connected by the DNA ligase, and that mismatch can be repaired. And so it depends on where the mismatch is located, and the cleavage site can be upstream or downstream of the mismatched region. And if it is downstream, a different set of enzyme will come in and remove a segment of DNA. And then the DNA polymerase 3 can come in and they synthesize a new segment of DNA. So this is how mismatch in DNA can be repaired. And actually, these are the bacterial enzymes. In human cells, we have homologs of MUT-S and MUT-L, but we do not have this enzyme MUT-H. So the mechanism of how human cells repair mismatch is still a mystery. So the last topic I will cover today is about DNA recombination. And this is a very interesting topic. And you see, this is the process of cell division. And at a certain stage, each of the chromosome have 4 copies. And then you see each of the 2 of these, they actually have a chance because they're homologous to each other. And then they have a chance to actually interact and they exchange information between them. And then eventually you see those, like when the cells divide and then this chromosome get into each of the haploid gamete cells. And so if you compare this one and the original one cell, and then you see there's some different, like the DNA actually recombination happened. And this is the electron microscope image showing like the chromosomes actually have a lot of crossover points, and they can actually change their information in this process. So this is the homologous recombination of DNA actually happens through this, what we call a holiday junction. And you see, this is what actually is this. It is this 4-way DNA. You see, these are 2 homologous DNA, and then when they get very close to each other, they actually this 2 double strand can actually interact with each other. And one strand from one double strand can evade the other double strand. And then eventually they form this holiday junction. It is a 4-way junction of 2 double helical DNA. And so eventually this 4-way junction have to be resolved by a set of enzymes in the cells, depending on how they get resolved. And then you see the possibility you end up with some copies of the DNA which looks similar to the original, but others you see the exchange between the blue strand double strand and the red double strand. You see this one, the blue strand double strand is not connected to the red one, and the red one is connected to the blue one. So that is a homologous recombination. It allows 2 homologous DNA to exchange fragments of information. And so this process is actually very well studied. And the first thing involved is to generate a single-stranded DNA from a double strand. And for this process, there's an enzyme called RAC-BCD. It is involved. RAC-BCD is an enzyme. It can cut the double-strand DNA from one end by removing nucleotides. And so when this enzyme recognizes a certain sequence called a chi sequence, and then it will stop. It will not cleave this strand anymore, and then continue to cleave the other strand. And then the result is it will generate one of these single-stranded DNA, which can associate with this RAC-A protein. So the RAC-A protein actually is very important. It mediates the strand invasion between one double-strand DNA to another double-strand DNA. So once this single-strand region gets generated, it can invade another double-strand DNA, which have a homologous DNA sequence, and eventually result in the formation of this 4-way junction called the holiday junction. So here I show you some of actual microscope image of the polymer of the RAC-A, and you see these are RAC-A polymers, and it can recognize the single-strand DNA and double-strand DNA, and allow the single-strand DNA to invade the double-strand DNA. So the 4-way junction of the holiday junction eventually have to be resolved by this nuclease. And you see, this is the structure of the IUVA protein. It is supposed to recognize the 4-way junction of the double-strand DNA. And eventually the IUVC, it will involve in the cleavage of this double-stranded 4-way holiday junction. And so here actually, this is the electron microscope image and the model of the RUAB covalent junction complex. And you see, this is the one of the arm which span to the RUVB. And RUVA actually recognizes four-way junction. So this DNA, homologous DNA recombination can be involved in DNA damage repair. For example, if the one of the double strand is damaged, which could have a nick on it, and then this replication, you see, if the replication fork replicates this DNA, and then it will stop at this point once it meets this gap region, and then the like by the action of RAC A and RAC BCD, and it causes the, like use one of the double strand as template, and then the other double strand will actually form a single strand and invade this double strand, and then eventually form a Howard junction. And after reservation of the Howard junction, the replication fork can be resumed. And similarly, this mechanism can be involved in the repair of double-strand DNA damage. For example, in human cells, we have two copies of the genes. For example, in the replication process, if one of them is damaged and one double strand is broken, and then they actually, through this homologous recombination process, they actually, the damaged strand can invade the good strand and form two Howard junctions. And eventually, after the Howard junction got resolved, and then the double strand DNA damage is repaired. So this DNA damage repair have very important implications. And here I show you one of the very interesting example, which is the gene editing by the CRISPR-Cas9 system. And so this is actually originally a mechanism by which the virus, for example, bacteriophage, they actually, bacteria can fight against the infection of phage, because if they have infection previously, they can actually store, bacteria can store some of the genetic information of the bacteria, of the virus in their genome. And then those DNA fragment can be transcribed into RNA, and then it will associate with this tracer RNA. And then the next time, if there's infection, this tracer RNA, which associated with the guide RNA, actually it can recognize double-strand DNA from the virus, and then cleave that double-strand DNA. So once that double-strand DNA from the virus is cleaved, and then the virus cannot replicate. So this is pretty recent discovery, and it's in the study of how the bacteria, bacteria fight against virus. And actually, Jennifer Doudna in UC Berkeley and Emmanuel Chapelier, and she is, I believe, in the Max Planck Institution. They actually studied this for a couple of years, and they actually come up with the idea of using this enzyme to get the modification of DNA. And so the one approach is that they can use this enzyme. For example, you can transfect the cell with the Cas9 gene, and then you transfect a plasmid coding for the guide RNA, and then you cut a certain DNA sequence in the genome. Through this non-homologous end join process, we did not talk about this. This is pretty like for double-strand DNA breakage, this is the error-prone repair mechanism. And so which is repaired, it may, for example, insert a nucleotide or insert a few nucleotides, and then it will cause a radial frame shift, and then like essentially cause the tag gene to be cannot be transcribed properly, translated into a functional protein. So that is the one function of the CRISPR-Cas system, and you can cut a specific point of DNA and then disable that gene encoded by the DNA. And another mechanism of gene editing is that if you can use the guide RNA to get the cut of the cleavage of the double-strand DNA. And at the same time, if you provide a piece of DNA, which have, for example, some other information in it, and through the process of homologous recombination, those information can be inserted into the target DNA. So these are some of the applications of the CRISPR-Cas system. And if you pay attention, you may already know Emmanuel Charpentier and Jennifer Doudna. They got Nobel Prize for chemistry this year for developing this technology. And this is very useful. It has been like a game changer in the molecular biology field, and everyone's using it to modify genes, especially for mammalian cells. For the bacterial cell DNA modification, it's pretty straightforward, but for the mammalian cell genome modification, it's very complicated. So they developed this technology which allows modification of the genome. Is there any questions about this? I think, yeah, look at your textbook, it have include this a lot of details about this, and even other potential applications. So if you are interested, you can read the textbook. So this technology can also be used to generate genetically modified animals like mice, which can be used in the research labs. And so when we talk about the immune system, we talk about how the diversity of antibodies can be generated by VDGA recombination. And here, this is you see those are the gene fragments, and they can be spliced out, connect to each other, and they contribute to the diversity of the antigen binding site of antibodies. And so this process is mediated by two enzymes called REG1 and REG2. And this enzyme can recognize specific signal sequence for splicing those fragments. And you see that the sequence in the middle of the two signal sequence can be cleaved, and then the two ends of the double strand can be connected to each other. So that is how this is still pretty active field of study in the last couple of years, and the molecular mechanisms just start to emerge. And here I show you, this is a structure of the REG1, REG2 recombinase bound to the double strand DNA. And in 2015, and there's a group in Harvard University, they actually used the electron microscope to resolve the structure of this complex. And there's still a lot of study, follow-up study about this. I recently attended a meeting and they showed some more details of other enzymes which have similar function. So before we end, I will give you a brief overview about how the homologous recombination can be used to generate transgenic mice. This is the technology before the CRISPR-Cas system, and it's purely depend on the homologous recombination. And you see, this is the transgene, and you can put it into a plasmid, and you can transfect the embryonic stem cells which have that gene, and then through the homologous recombination, this, like for example, this neo marker can be inserted into one of the exons of the gene. And then eventually this modified embryonic stem cell can be injected into a mice. Those stem cells can be injected into an embryo. And then those embryo put back into the mice. And eventually the mice, because this embryo, when they develop, some of the cells have the transgene, modified gene, some of them don't, and eventually they give birth to this kind of chimeras. And so if you cross those chimeras, and because some of them have one gene allele got modified, and some of them have another, and then eventually you have a chance to get homozygous, which have both of the gene modified. This takes a lot of time, a lot of money, but these days, at this stage, this gene can be modified by, for example, the CRISPR-Cas system, and then the embryos can be put back to the mice, and then they can be selected and then generate gene knockout mice. It is a lot faster and it costs less. So that is pretty much what we have today. And before we end, I'll show you a video about the gene editing by CRISPR-Cas technology. And it's a very nice video, and I'll show you. Oh, see, where's my video? It should be somewhere. I'll see which one is my video. Oh, it should be this one. Oh, okay. Now this is
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAN. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
That's pretty much we have for today, and we still have a couple of minutes if there are any questions about the topic today. We can have a little bit question and answer session.
I have a question.
Uh-huh.
So is there any a human equivalent protein similar to the Cas9 that
No,
can...
you not need to use, there's no human protein, those are the bacterial protein. You have to, like if you want to do gene editing, you have to introduce Cas9 and the guide RNA into the human cells.
Oh no, I was wondering if there was any similar protein that would stop other viral infections like retroviral infections.
No, it's a bacterial system.
So only in bacteria?
Yeah, this is the bacterial antiviral response. It's not involved, it does not exist in human. So we do not have a Cas9 gene.
Thank you.
It's a good question. Is there any other questions?
Dr. Lee,
Could
you go back over the DNA methylation and mismatch repair? So what exactly is the problem, I guess, or the mismatch within the DNA?
Let me share the slides and let me know which one you're talking about. What is you are talking about?
It's the DNA methylation and mismatch repair.
Yes,
that one.
Yeah. So the bacteria have a enzyme called dimethylase. It will actually modify the newly synthesized strand with methyl group. So before this modification happens, any mismatch can be repaired because the cells know which one is the original, which one is the new strand.
Okay, so it's looking at which, so in the very first one, that's what we want, correct? We want the two methyls.
So this mutyl S and mutyl L, it will actually bind to the mismatched region, and then this DNA loop out, and then until it recognizes that sequence, which contains one methylated adenine and the other one not methylated. And then it will cut onto the one not methylated. That is how it works.
Oh, okay. And then it'll put a methyl back on that, or just
no,
repair?
it will just remove this segment. Once it is cleaved, then a segment can be removed. For example, here, that segment can be removed from the newly synthesized strand.
Yes, sir. Okay, that makes more sense.
Then the DNA polymerase will come in and synthesize another one here.
Okay, so there's just some kind of
because this is 5' 3', and it goes that way. Yeah, but it will fill up DNAs, and then eventually they can be connected to each other.
So there's some type of mismatch within that strand or at that certain spot.
Yeah.
Correct?
Yeah, it will recognize the sequence and then cleave the newly synthesized strand, which is not methylated. And then the enzyme will, like a polymerase, will lay a new strand of DNA here. And then the ligase will connect them together. That is how it works.
Thank you.
Any other questions? Okay, if there's no questions, we'll just stop here for today.